Corn smut is a plant disease caused by the pathogenic fungus Ustelagomates that causes smut on maize and tosandi. The fungus forms galls on all above ground parts of corn species, and is known in Mexico as the delicacy huitlacoc. It is eaten, usually as a filling, in quesadillas and other tortilla-based foods, and soups. Etymology: in Mexico, corn smut is known as huitlacoc, Spanish pronunciation, sometimes spelled Cuitlacoc. This word entered Spanish in Mexico from Classical Nahuatl, though the Nahuatl words from which huitlacoc is derived is debated. In modern Nahuatl, the word for huitlacoc is Cuitlacocan, Nahuatl pronunciation, and some sources deem Cuitlacoqui to be the classical form. Some sources strongly give the etymology as coming from the Nahuatl words Cuitlatl, excrement or erend, actually meaning excrescence, and cachi, cach equals to sleep, thus giving a combined mismeaning of sleeping, hibernating excrement, but actually meaning sleeping excrescence, referring to the fact that the fungus grows in between the corns and impedes them from developing, thus they remain sleeping. A second group of sources deem the word to mean raven's excrement. These sources appear to be combining the word Cuitlacoc for thrasher with Cuitla, meaning excrement, actually meaning excrescence. However, the avian meaning of Cuitlacoc derives from the Nahuatl word song Cuicatl, itself from the verb to sing Cuica. This root then clashes with this reconstruction's second claim that the segment Cuitla comes from Cuitla, excrement. One source derives the meaning as corn excrescence, using Cuitla again and maize tlali. This requires the linguistically unlikely evolution of tlil-maize into tlacoc. Characteristics: the fungus, itself, infects all parts of the host plant. In order to spread to new hosts, fungal tissue invades the ovaries of its host, causing the corn kernels to swell up into tumor-like galls analogous to mushrooms. These galls are made up of hypertrophied cells of the infected plant, fungal threads, and blue-black spores. These dark-colored spores give the cob a burned, scorched appearance, thus explaining the meaning of the generic name Ustelago, coming from the Latin word ustellare, to burn. Biology: life cycle. When grown in the lab on very simple media, it behaves like baker's yeast, forming single cells called sporidia. These cells multiply by budding off daughter cells. When two compatible sporidia meet on the surface of the plant, they switch to a different mode of growth. First, they send out conjugation tubes to find each other, after which they fuse and make a hypha to enter the maize plant. Hyphae growing in the plant are dikaryotic, they possess two haploid nuclei per hyphal compartment. In contrast to sporidia, the dikaryotic phase of Eumedes requires infection of the plant to grow and differentiate, and cannot be maintained in the laboratory. Proliferation of the fungus inside the plant leads to disease symptoms such as sclerosis, anthocyanin formation, reduced growth, and the appearance of tumors harboring the developing teliospores. Mature spores are released from the tumors and spread by rain and wind. Under appropriate conditions, a metobasidium is formed in which meiosis occurs. Resulting haploid nuclei migrate into elongated single cells. These cells detach from the metobasidium to become the sporidia, thus completing the life cycle. Host, pathogen conflict. Plants, in general, have evolved efficient defense systems against pathogenic microbes. A rapid plant defense reaction after pathogen attack is the oxidative burst, which involves the production of reactive oxygen species at the site of the attempted invasion. As a pathogen, MADAs can respond to such an oxidative burst by an oxidative stress response, regulated by gene YAP1. This response protects you, MADAs, from the host attack, and is necessary for the pathogen's virulence. Furthermore, MADAs has a well-established recombinational DNA repair system. This repair system involves a homologue of Rad51 that has a very similar sequence and size to its mammalian counterparts. This system also involves a protein, REC2, that is more distantly related to Rad51, and BRH2 protein that is a streamlined version of the mammalian breast cancer 2, BRCA2, protein. When any of these proteins is inactivated, sensitivity of MADAs to DNA damaging agents is increased. Also, mitotic recombination becomes deficient, mutation frequency increases, and meiosis fails to complete. These observations suggest that recombinational repair during mitosis and meiosis in MADAs may assist the pathogen in surviving DNA damage arising from the host's oxidative defensive response to infection, as well as from other DNA damaging agents.